Welcome everyone. So today we will discuss about bacterial cell structure, and believe me, it's very important topic in terms of GAT-B. Okay, so without wasting a single minute, we will see what bacteria is. I hope you know what bacteria actually is. So just take a quick look on bacteria. Okay, so bacteria are prokaryotic organism, as all of us know, and characterized by absence of a true nucleus and true membrane-bound organelle. That's the main difference between the prokaryote and the eukaryote, right? And their cellular organization is simple but highly efficient. Yeah, that's very important word actually. The bacteria are very tiny but are very smart, right? Because whatever we are doing, let's say we are producing our protein, like our cell are making our protein through translation and all these things, bacteria is also doing the same thing in a very smart way. Some general features like bacteria size varies from 0.5 to 5 micrometer, and the genetic material is naked DNA. Okay, so bacteria divide by binary fission, and the ribosome is 70S. In case of prokaryote, it's 80S. Okay, and the cell wall, bacteria has cell wall that made up of peptidoglycan, except mycoplasma. That you need to remember that mycoplasma don't have any peptidoglycan layer, right? Now, depending on their organization or shape, we can divide them mainly in major three classes. One is the cocky, that means mostly the spherical. Now, if single is there, that's called coccus, and then if it's two spherical bacteria attached, that's called diplococci, right? Followed by that, if there's a particular chain, it's called the streptococci, and if this is a cluster, then it's called the staphylococci. And if you see the bacillus, bacillus is kind of rod-like things, right? And if multiple rod-like things is clustered together, that's called the streptobacilli, right? Similar thing, we have some other bacteria like Vibrio cholerae, that's the common shape bacteria, right? Some spirochete, spirilla, that's spiral shape, right? So if we consider the bacterial shape type, mainly we have two type, cocci and bacilli, and except these two, we have also some irregular shape, that's Vibrio, maybe spiral, maybe spirochete. Okay. Next. So that's the general structure of bacteria, right? So if we start with the outer layer, okay, let me change the color. Yeah, that's fine. If we go with the outer layer, in the outer layer we have lot of surface appendage, right? Flagellum, right? And then pili is also there, right? So that's the most surface, right? Then what we will encounter? We will encounter with the slime layer if bacteria has anything, let's say some capsule or any other thing, right? Slime layer. Followed by this, bacteria has cell wall that's made up of the peptidoglycan, PGN. Okay, we will see how PGN is synthesized, or maybe how PGN are connected. Next, the cell membrane, that's the regular plasma membrane made up of phospholipid. Okay, and then as a genomic material, bacteria has DNA, that's the bacterial own genomic material, as well as the plasmid, right? The main function of the plasmid is that it provides a lot of antibiotic resistance, maybe some toxin production and all these things, right? And bacteria has some cytoplasm. Let's say this is the cytoplasmic area. Here you can see some kind of black dot. Those are actually some storage molecule, right? We'll see all of the storage molecule as well. Bacteria has ribosome, that we already know that it's the 70S ribosome. Okay, next. Now we'll discuss each and every thing of them in detail. So if we consider the external structure, that's a glycocalyx, the outermost covering, that can be a capsule or a slime layer, right? Capsule is more organized, that you need to remember. Capsule is more organized and it is more firmly attached, whereas the slime layer is loose and unorganized. So the main difference between capsule and slime layer, that means let's say this is the bacteria and that's the peptidoglycan, and above peptidoglycan there is one particular slimy things made up of mainly carbohydrate, right? That's called the capsule or slime layer. The main difference is capsule is more organized, okay, and densely packed, and while slime layer is loose, not properly organized. Main function is that protection from the phagocytosis. Okay, prevent desiccation and biofilm formation. That's very important question. People can ask you which of the following is participating in the biofilm formation. That's the glycocalyx, mostly the capsule and the slime layer, right? And also virulence factor. The main thing is the protection. Let's say if some phagocytic cell is coming to grab the bacteria, the slime layer is shielding the bacteria, right? It can just prevent those immunogenic cells to recognize the bacteria. Next, flagella. It's very important thing, and just think about that how smart bacteria is. It has some flagella, and with the flagella it's just moving here and there, right? So in the figure also you can see it's a long flagella, right? Some whip-like appendage. And if we just go into this detail, right? Let's say this is the okay, we'll see in the gram-positive, gram-negative bacteria. But as of now, just remember, in case of gram-negative bacteria, okay, there are two membrane, outer membrane and inner membrane. In between, peptidoglycan is there. So here, it's the gram-negative bacteria. In case of gram-positive bacteria, only one cell membrane is there, and followed by this is the peptidoglycan, thick peptidoglycan. So that's the gram-positive bacteria that we will discuss in detail. So here you can see here one plasma membrane, two plasma membranes are there. That means this is a gram-negative bacteria, and we will see the arrangement of flagella in gram-negative bacteria. So this is a thin peptidoglycan layer, and here this is a type 3 secretion system, and different things are present here. And the flagella is made up of mostly 3 to 4 rings. Let's say you can see the MS ring, the P ring, right? That means just to rotate the flagella, it needs some socket kind of things, right? So it has multiple ring. With this ring, it just rotates, right? And there's also some meaning of this rotation. If there is clockwise, it is indicating one thing. If it's anti-clockwise, then also it's indicating different thing, right? And yeah, remember. And then this is the hook-like filament. The flagellum actually can sense temperature. Flagellum decide the actually motion of the bacteria, right? Now the flagella can be one, the flagella can be multiple. Okay, based on this we can divide the bacteria in a few of these categories. First one is the monotrichous. Monotrichous means only one flagella at one end. Lophotrichous, that means tufted flagella at one end. That's very important in terms of exam. Okay, so lophotrichous means tufted flagella. Let's say 5 flagella at a single end. Amphitrichous, that means it's in the both end, but only in the end portion. Let's say here 1, here 1, here 3, here 3. But peritrichous means across the surface. It has multiple flagella. So peritrichous means across the surface. Multiple flagella. And monotrichous means only one flagella, right? So depending on the arrangement of the flagella, these are the following category: monotrichous, lophotrichous, amphitrichous and peritrichous. Monotrichous means only one flagella at one end. Lophotrichous means multiple flagella, tuft of flagella at one end. Amphitrichous means at both the end. Peritrichous means over the surface, right? Then comes the pili or fimbriae. So those are the small appendages. Let's say this is the bacteria, and let's say this is the flagella, but pili will be quite shorter, right? So pili or fimbriae, so it can be either fimbriae, that attachment to the surface. So the main role of the pili is this, the attachment with any kind of surface. Let's say this is one glass surface, or can be any plastic surface. Now bacteria want to adhere here. So what the bacteria will do, bacteria will just need the help of the pili. And another thing is that sex pili, that help in conjugation of DNA transfer. If you see here, let's say this plasmid is here, okay, so we can say A positive. This plasmid is not here, we can say A negative. So what will happen, it has pili, it has pili. These two pili is making one tube, sex conjugation tube, right? And through this conjugation tube, the plasmid is transferring, right? So what can, let's say it has the kanamycin resistance gene, right? Kanamycin resistance, but this strain doesn't have the kanamycin resistance. But if the transfer happen, then what will happen? What will be the fate? The fate will be, this bacteria will also have the kanamycin resistance, right? After the conjugation. So that's the role of the sex pili. Next, cell envelope. First thing comes in our mind, that's the cell wall. Of course, cell wall means, let's say this is the bacteria and this is the thick cell wall. If it is a gram positive bacteria, here we will discuss, right? It's made up of peptidoglycan and peptidoglycan. So peptidoglycan is composed of two things, two of the sugar moiety, right? One is the N-acetyl glucosamine and another is the N-acetyl muramic acid. N-acetyl glucosamine, that's NAG. NAM means N-acetyl muramic acid. Those are connected through beta-1,4 glycosidic linkage. Please note that those are connected by beta-1,4 glycosidic linkage. I hope you know what is the role of glycosidic linkage. So it just connect two of the sugar molecule, right? And release some water molecule. That's the glycosidic bond. Okay. So based on this cell wall, we can divide bacteria in two types, gram positive, gram negative. In case of gram positive, remember the peptidoglycan is thick. Thick means there is only one layer, one cell membrane, right? And after this, the thick membrane is there. In case of gram negative, we have two membrane, outer membrane, inner membrane, and inside a thin peptidoglycan layer. So in case of gram negative, it's thin. As it's thin, that's why it can easily lose the staining, and you can stain it with the counter stain. That's how the gram positive and gram negative name came, right? So here outer membrane present. In case of gram negative bacteria, another thing is the lipopolysaccharide, LPS. So remember, LPS is present only in gram negative bacteria, not in gram positive bacteria. So what is the component of LPS? Lipopolysaccharide composed of lipid A, that's the endotoxin, core polysaccharide, and O antigen. So in the O antigen, there is some antigenic variation, and that actually creates some variation among bacteria, right? So LPS has three components: lipid A, that's the endotoxin, core polysaccharide, and O antigen. O antigen means it has some carbohydrate moiety, and maybe there is some variation in the O antigen across different pathogenic or virulent strain. Next, if we see the gram positive things, then it has the teichoic acid, it has the lipoteichoic acid, right? And it doesn't have any outer membrane, it has only one membrane. And gram positive bacteria is more sensitive to penicillin. So what penicillin do? Penicillin inhibit the transpeptidation. Penicillin inhibit the transpeptidation. Let's say, if you see the procedure of peptidoglycan synthesis, there are 5 amino acid, right? So let's say if it's one peptide, carbohydrate chain, it's another peptide, carbohydrate, carbohydrate chain, 5 peptide, carbohydrate chain, 5 peptide. Now they will make some linkage here, right? So the transpeptidation is inhibited by the penicillin. So gram positive bacteria are more sensitive to penicillin, right, rather than the gram negative bacteria. Next, cell membrane. So cell membrane, it is a phospholipid bilayer, so it lacks the sterol except the mycoplasma. So see, this is the peptidoglycan, this is the plasma membrane, right? So the plasma membrane function is same. The main thing is that the bacteria, this is a gram negative bacteria because it has two membrane, right? This is the inner membrane, this is the outer membrane, and this is the periplasmic space, right? So what's the role inside the periplasmic space? All the ATP synthesis like electron transport system occur, in this particular space, periplasmic space. Write down periplasmic space of gram negative bacteria, right? And in gram positive bacteria also there is some space in between. There the ETS happen, right? Transport means bacteria also need some nutrient, right? Carbon, oxygen, nitrogen. So all this molecule will be transported across the membrane, and this is selectively permeable, right? Some bacteria has acquired antibiotic resistance because they have some antibiotic efflux pump. Let's say if your bacteria has antibiotic efflux pump, if they consume kanamycin, they can efflux it, and then they will be again resistant to the particular antibiotic, right? Remember, this bacterial or prokaryote cell doesn't have the sterol except the mycoplasma. That's quite important. Next thing is the cytoplasm, cytoplasmic component. See, this is the cytoplasm inside. What's there? There is some ribosome, there is a nucleoid, there is some plasmid, and there is some storage granule. Let's consider about the cytoplasm first. It's a gel-like matrix that's similar, and it contains a lot of enzyme, right? Let's say all the glycolytic enzyme and all the necessary, because bacteria don't have some separate compartment, right? So whatever they need to perform, everything will be done in the cytosol or cytoplasm only. Then nucleoid. Nucleoid means the irregular region containing the DNA. That's the nucleoid. Nucleoid. It can be single, circular, or double-stranded DNA. No membrane, no nuclear membrane. So in case of eukaryote, there is a nucleus and then there is a membrane. But in case of prokaryote, nothing is there. It is just the naked DNA, right? So supercoiled DNA stabilized by RNA and protein, right? There are a lot of histone kind of protein like IHF, HNS. This kind of protein are there. This is a histone-like protein. This protein helps in supercoiling of the nucleoid. Remember, HNS and IHF are histone-like protein. Histone-like protein that help in supercoiling. Next thing is the plasmid. Plasmid is small, circular, extra chromosomal DNA. So that's the plasmid. So what's the actual role? Not essential for survival, but it provides actually a lot of benefit. Let's say it provides the antibiotic resistance, it provides the virulence factor as well as some metabolic trait as well. Let's say if you add some kanamycin, and if the bacteria has a kanamycin resistant plasmid, right, then what will happen? The bacteria will survive. So it's just for survival of the survival means some extra benefit of the bacteria, but not essential. Next, ribosome. Eukaryotic ribosome is 8 Ts, but the bacterial ribosome is 7 Ts, right? Ribosome that help in protein synthesis, they just scan the mRNA. Bacteria has signed Dolgarno sequence, right? In the mRNA, signed Dolgarno sequence. Just remember what is SDS. This is signed Dolgarno sequence the mRNA has, and then the ribosome starts scanning it. Okay, so if you are tetracycline or streptomycin, they will block the ribosome attachment and the protein synthesis part mostly, right? So 70S ribosome, if you can classify it, it's 30S and 50S. Among 50S is 23S and 5S, and amongst 30S it's just the 16S, right? That's all about the ribosome. Then comes the inclusion bodies. What's inclusion body actually is? That's the stored things. Let's say bacteria need to store something, right? Let's say bacteria want to store glycogen, so just for energy. So some molecule, let's say this is the bacteria, and nucleoid is there, plasmid is there, ribosome is there. Now some dot thing you will also find. They are storing something like polybutyl hydroxybutyrate. This is a carbon source. Glycogen for energy purpose. Volutin for phosphate. Right? Sulfur granule just for incorporating the cysteine amino acid there. Right? Next, the spatial structure. So the thing is endospore. Endospore means their dormant and resistant structure. Let's say whenever some bacteria is facing some kind of stressed condition, it can divide. At that time, they will make the endospore. So what's the role of endospore? Endospore will create such a environment that the bacteria can grow in any time, right, through this endospore. So mostly formed by two kind of genus of bacteria. One is Bacillus and another is a Clostridium. Please remember Bacillus and Clostridium. Okay, so there are few layers. There is a core, cortex, spore coat, and exosporin. Right. So mostly they provide resistance to heat, radiation, chemicals, as well as desiccation. Right. The endospore. Remember, the endospore has one acid called dipicholinic acid. Very very important. So the dipicholinic acid makes some complex with calcium that actually provides some extra stability to the endospore. So dipicholinic acid DaCa2 complex provides stability. That's for stability of the endospore. Stability of the endospore. Right? Endospore are not reproductive structure. So let's say whenever some bacteria is facing some issue, right? At that time what the bacteria will do, they will start making the endospore, but not all the bacteria, mostly few genus. Let's say as example Bacillus and Clostridium. Next, there is some difference between gram positive and gram negative bacteria, right? In case of gram positive bacteria, if you see the peptidoglycan, it's thick, and in case of gram negative, it's thin. So here is a clear difference. This is gram positive, peptidoglycan is thick, and this is the gram negative, peptidoglycan is the thin. So this is gram negative, this is gram positive. In case of gram positive, outer membrane is not there, right? In case of gram negative, there are two membranes, inner membrane and outer membrane. Teichoic acid present only in case of gram positive bacteria, not in gram negative bacteria, right? In gram negative, teichoic acid is absent. The LPS is present in gram negative bacteria. LPS means lipopolysaccharide that has three components: lipid A, core antigen, and O, right? Those are present in the gram negative bacteria, but absent in gram positive bacteria. So if you remember few of these things, you can expect at least one or two questions in the GAT-B, right? Okay, so that's all about today. Thanks.